When human sequence variation is significantly associated with a disease or trait, that can give us clues about the genes and pathways that underlie that phenotype. Rare variants can be especially informative because they often have much larger effects than common variants. But because these variants are rare, single variant genetic associations don't have much statistical power. Instead, gene-level associations, calculated from the aggregate burden of rare variants in a gene, can suggest correlations of genes with diseases and traits. The Genetic Association Interactive Tool, or GAIT, securely accesses protected individual-level whole exome sequence data to allow you to do these calculations on the fly. This video will show you how to use the interface. For all the details about the data and the underlying methods, please see our online written documentation. You can find it by clicking Help in the upper menu. You can access GAIT from the Tools menu of many knowledge portals. We'll demonstrate here with the Common Metabolic Diseases Knowledge Portal. GAIT accesses associations for a defined set of cardiometabolic traits and is only available in the knowledge portals that include those traits. At the top of the page, you'll find a roadmap that illustrates the overall workflow of the tool. There are also links to the full documentation and this video tutorial. To use GAIT, first enter the name of a gene of interest and select the name from the list. Optionally, you can select an individual transcript of the gene to analyze. Next, select a data set. If you choose 52K, which refers to the exome sequence analysis from the AMP T2D Genes Consortium, you'll be able to perform aggregation tests for type 2 diabetes and 23 additional related quantitative traits. If you choose TopMed, referring to the exome subset of whole genome sequencing from the TopMed Consortium, you'll be able to run tests for type 2 diabetes, fasting glucose, or fasting insulin. Next, to retrieve the set of variants that will go into your test, select a mask. Masks are filters that group variants based on their impact on protein function, as predicted by different combinations of bioinformatic methods. In general, you want to create a set of variants in which most or all of them affect function of the protein. The masks are ordered from the most stringent at the top to the least stringent at the bottom. In other words, if you select LOFT, you will get a smaller set of variants that are likely to cause a loss of function in the protein, while if you select the masks further down in the list, you'll retrieve variants with less severe predicted impact. All of the methods used in each mask are listed and referenced in our GAIT help documentation. Go to the Help menu, click on GAIT under Tools, and scroll down to see the list of methods used to create the masks, and also the composition of each mask with these methods. After selecting a gene, optionally a transcript, a dataset, and mask, click the Search Variants button. You'll see the list of variants that meet your criteria. You can now refine the list by deselecting or selecting individual variants if you wish. For example, you could edit your set of variants so that it only includes variants located within a domain of a protein that is known to be important for function. You can see an example of this use of GAIT in our rare variant gene-level associations workflow. Then select phenotypes, as many as you like, and one or more burden test methods. Finally, click Run Analysis. The results are presented in tables. Be sure to check out our help documentation for all the details of this analysis.